Good afternoon, everybody. Welcome to the first MHRC seminar of our fall semester. It's a great pleasure for me to introduce our speaker today, Dr. Charlotte Peterson from the University of Kentucky. And Dr. Peterson, we've been trying to get Dr. Peterson to come to York University in Toronto for quite some time. And of course, COVID didn't allow that. And so we're doing the next best thing here by having a Zoom meeting with her. And she's, I think, happy to be here. Charlotte is the Joseph Hamburg Endowed Professor and Director of the Center for Muscle Biology at the University of Kentucky in Lexington, Kentucky. And she's just been describing her muscle center, and we all know how active that group is. And it's just a great pleasure for us to have her here because of the really pioneering work that she's been doing on satellite cells, and for us muscle biologists, you know, to understand how satellite cells operate during growth and development, and also during hypertrophy. And their contribution to sarcopenia is just really, really fascinating, and of course controversial. So there's a great field out there, but Charlotte is really leading the way to our understanding of what satellite cells are doing. So without any further talk from me, I'd really like to have Charlotte please turn on her camera and maybe share the screen with us, and take it away, Charlotte, and tell us about your topic on satellite cells, exercise, and aging.
Okay, make sure I'm not muted, which I could be talking away and you wouldn't hear me. I hope that's okay. And I want to thank David first for the invitation. And I so regret, or I'm sorry, that I'm not up there in Toronto with you all. I was really looking forward to this trip. But as you say, this is the next best thing, because I love to share our work and tell you about the latest that we have regarding primarily satellite cells, but a couple of other little tidbits along the way as well.
Sorry, Charlotte, I'm just going to interrupt. Could you just stop? We're seeing a kind of a different view right now for your PowerPoint. Could you just stop sharing it for a minute and try that again, please?
Okay, so are you saying the the version?
Yeah, presenter view.
Yeah.
Thing, yeah.
Okay, let me see if I can. Okay, stopped sharing. Let me go here and see if I can move this, because that's perhaps what was happening with David's as well, because we were seeing his cheat sheet version. No. Hide. How about if I hide the viewer presenter version? Do you see it now? Is it on the right screen?
You just need to start sharing your screen again, please.
Oh, sorry. Better?
Perfect.
Okay. So technical issues are always a challenge, aren't they? Okay, so I'm going to dive right in and give very little background, given that, you know, you're very impressive Muscle Health Research Center. I know I don't have to give much background on muscle structure, but I always like to start with okay, but now I'm not able to advance my slides. Boy, this worked fine when we did our there. How's that? Can you see my next slide?
Working great.
Okay, fine. So I always like to just give this, just this brief introduction to satellite cells, because many people outside of the muscle field don't understand or don't appreciate them, in that the muscle fiber, of course, is this big, long, syncytial fiber. And I'm going to see if I can do a pointer options, laser pointer. There we go. So you have, of course, the muscle and the big fascicles, and then if you go down further in magnification, you see this beautiful single cell muscle fiber that is then further organized into myofibrils and the sarcomere. But the cell that, as David pointed out, I'm going to talk a lot about today is the satellite cell, which is tightly associated with the muscle fiber underneath the basement membrane. And this is, of course, the adult muscle stem cell. And in the last several years, 10 or 12 or 15 or so, they've been very intensely studied, and it's been largely possible to the development of an antibody against the protein, the transcription factor PAX7, which here I'm showing you a couple of images from Pete Zamet's paper, where you're seeing the satellite cell fluorescing green due to a fluorescent PAX7 antibody. And it's really been a powerful tool, since in muscle, PAX7 is absolutely satellite cell specific. And here, just showing on an isolated single fiber, and here in the cross section, here pink, you can see the labeled satellite cell. Now in our lab, I just wanted to give an example here. They're very easily to identify and count using PAX7 immunohistochemistry. And so a lot of times I will show you data about the number of satellite cells per fiber, and we count satellite cells. And I just wanted to see with the staining protocols that we've worked out, they're very rare, they're stem cells, but they're easily quantified. So what do we know about satellite cells? That it was speculated for many years that they are the muscle stem cell. And what we do know by both in vivo and in vitro experiments, that they really are the primary cell that's responsible for muscle repair and regeneration following injury. And that's just illustrated here in a cartoon from one of Grace Padlock's work several years ago, to just illustrate that you have a muscle, again, the basement membrane around it with the satellite cells, actually very much indented, closely associated with the muscle fiber. If you have injury, they proliferate, they migrate to the site of injury, they fuse, and they can reform either the entire myofiber or the section of the myofiber that's been injured. And the hallmark, as I'm sure most of you know, of a regenerated muscle fiber is these central nuclei, because until they mature and the myonuclei can push to the periphery to allow the sarcomere to form, centrally nucleated fibers are generally considered a hallmark of a regenerated fiber. So this we've known for a long time, and this is primarily what the field of muscle field and satellite cells field studies with regards to satellite cells, because they are clearly required for muscle regeneration. But what I want to talk to you today about what we consider perhaps more physiological models and a more physiological role for satellite cells. And first, I'm going to talk to you about our work. We focus very heavily on hypertrophy, and here it's clear that satellite cells can fuse into a growing muscle fiber, add nuclei as it's growing. And so I'm going to spend a great deal of time talking about this, and then a little too relevant to muscle homeostasis across the life of the animal and into old age. So what we got when we got started on this, we were very much influenced by the myonuclear domain theory, which states that the myonuclear domain remains constant during muscle growth, which requires the satellite cell to fuse in to donate a nucleus to maintain a constant nuclear to cytoplasmic ratio. And this theory has been around for many years, and it's sort of illustrated here just to make it incredibly clear. And first here, we've just talked about regeneration. So here you have satellite cell activation, proliferation, differentiation. They can form a new myofiber. But in the case of a hypertrophic stimulus, they can be activated, proliferate, but then they can fuse into an existing myofiber to facilitate growth. And then, of course, a subset of them go back into the quiescent satellite cell state against the muscle fiber. And the myonuclear domain theory posits that here you have a myofiber, and if it's going to grow, you see that the actual area of the slice of pie stays the same because you add new nuclei from satellite cell fusion. So when we started our studies here on muscle hypertrophy almost 10 years ago, we started with the hypothesis, well, muscle hypertrophy will not occur then in the absence of satellite cells. And luckily, models became available, genetic mouse models, which allowed us to specifically deplete satellite cells so that we could directly test this hypothesis. And these are the mice. They're conditional genetic mouse models. And I'm going to explain this just briefly. I'm sure many of you work with some of these Cree mice. And the mice that we've worked at that I'm going to talk about for the first half of the talk are in which we crossed a mouse that we obtained from Gabby Cardone in which the Cre recombinase, so that's a viral enzyme that encodes a recombinase, is knocked into the PAX7 locus. And as I told you, the PAX7 promoter is absolutely satellite cell specific. Now, this is a modified Cre with a modified estrogen receptor, so it's not active. Its recombinase activity is not active until you treat the mice with tamoxifen. So this is the first mouse, and we crossed this to another mouse in which a very strong constitutive promoter, the ROSA26 promoter, was driving the diphtheria toxin A gene. However, there's a stop codon, so that under normal conditions, the DTA, this toxin gene, is not expressed. However, if you cross these mice, treat them with tamoxifen, activate the Cre recombinase, this stop codon is flanked by 2 LOXP sites, which are the sites recognized by the Cre recombinase that recombines out this stop codon. So now you have PAX7 DTA. DTA is expressed at high levels, and it kills all the satellite cells. So this is the mouse model, and what I'm going to talk to you about for the most of the talk. We work with fully grown adult mice, 4 to 6 months of age, so long bone growth is complete. They're fully grown. I'm going to show you one slide where we've gone back and tried to look at immature, younger mice, which many people work with in the field. And here is our tamoxifen regimen. So we give 2 milligrams a day for 5 days with intraperitoneal injection, and to effectively activate the Cre recombinase and do the rearrangement, the genetic recombination. And then for all of our experiments, we wait at least 2 weeks, sometimes longer, couple of months, to really remove all the tamoxifen from the system. We want the tamoxifen to be gone. There's no artifact in terms of tamoxifen effects on the mouse. And that in our studies, that we really only include in our studies those mice that are really effectively depleted. Sometimes the tamoxifen, if the mice are fatter and the tamoxifen is taken up in the fat, that the satellite cell depletion is not effective. But all the experiments I'm going to show you, and in the slides they'll be called satellite cell minus, means the ones that are treated with tamoxifen and are effectively depleted. And we test always first, the assay that we do to show then functionally that they're depleted is that we inject the tibialis anterior with barium chloride, which causes massive degeneration. And then we wait different periods of time. And here you can see a PBS injected vehicle animal. And after a week of following barium chloride injection, you see all these nice little regenerated muscle fibers. Things are looking good. This is the satellite cell depleted animal, and you see that regeneration is completely impaired. It's severely impaired. So we know that satellite cells are functionally depleted. And that's sort of our, as I said, we count the satellite cells, but then we do this functional assay in the tibialis anterior to demonstrate that they really are, that they're not there and regeneration is impaired. But because we're interested in hypertrophy, the model that I'm going to tell you about is that we use in the lab mostly is synergist ablation. And that's a surgical model of hypertrophy in which when it was first established, basically the whole gastric meatus and the little bitty dark red here soleus were completely removed, which caused hypertrophy, tremendous hypertrophy in the plantaris muscle. So the synergist muscle hypertrophy. More recently, and what I'm showing you here is that because that's such an incredibly harsh overload on the plantaris and actually does cause some damage, synergist ablation, if you remove these muscles, we now more or less basically do a tenotomy, but it serves the same function as activating the synergist muscle. So you see here, we remove usually a little bit of the muscle of the gastroc and a little bit of the plantaris and the tendon, which causes hypertrophy of the plantaris. And this is a very robust hypertrophy, and we use it more or less as a mimetic to resistance training. So it's a very strong functional overload of the plantaris muscle, and you can see that the muscle hypertrophies get a tremendous hypertrophy even within 2 weeks. Okay, so that's the model that we're using. What you can see in this model, the first thing I just want to demonstrate is that in fact, during hypertrophy, and this is 2 weeks, I say 2, of the plantaris muscle, you can see normally as the myonuclear domain theory would posit, and as many people have shown, including us, is you do see satellite cell fusion with hypertrophy and you get nice myonuclear accretion. In our tamoxifen-treated animals, that's completely eliminated. And we count the nuclei here, both on single fibers, and I'll show you some of those data, but in cross-section, this is a section which has been immunohistochemically stained with an antibody against dystrophin, which is in the sarcolemma, so that it allows us to identify DAPI-stained nuclei either within the muscle fibers or between them. And so these are either satellite cells or fibroblasts or macrophages or any number of cells, but we can specifically count myonuclei, which is how we quantify these images. So clearly, we've prevented myonuclear accretion. What we see is that you get significant growth still. So growth is not inhibited early on, that is. So here, when most of the initial growth happens, 1 week and 2 weeks, you see that without satellite cells, you get significant growth, which is in no way negatively impacted by the depletion of satellite cells. But over time, if you let the animals go long enough, here 4 weeks and 8 weeks, you see that growth is blunted. And this is shown in wet weight of the muscle normalized to body weight. And here we're showing fiber cross-sectional area, where you see, which of course is not as dramatic as the wet weight, you see a nice increase at 8 weeks. The hypertrophy has occurred, and that there is hypertrophy at 8 weeks in the satellite cell depleted muscle, but it's significantly blunted. So what I'm going to take just a little aside here and step back. So all these experiments, as I've told you, are in mice that are 4, 5, 6 months old, so greater fully grown animals. There is controversy in the literature, as David pointed out, that others have reported using modifications of synergist ablation or other models of growth, like downhill running and other things, where they report there is no growth without satellite cells. And we looked carefully at those papers and we talked to the authors, and it occurred to us that, you know, in all cases, they were using mice that were younger than ours. They would use mice that were 2 to 3 months old. You might not think that's much different, but those mice are still growing. The long bone growth is not complete. They're still growing. So we went back and did a large cohort of mice, and we repeated our cohort at older mice, but then took some mice, and at about 2.5 months of age, we did our tamoxifen treatment, our washout, overloaded them, so we sacrificed them just over 3 months of age. And what you can see in those animals, again, cross-sectional area, increases in the young animals with 2 weeks of synergist ablation, but in the absence of satellite cells, you see no growth. And this is what many people have reported, that you don't see growth in muscles without satellite cells. However, in the full-grown adult animals, you do. So I think it's really important to note that that the age of the animals is really important, and that all of our work is in full-grown adult animals, and that one would get a different result if you used a 2.5 or 3 month old animal compared to one that's over 4 months of age. Okay, so that was just a little aside to address some of the controversy that is in the literature. Now coming back to our full-grown adult mice at 8 weeks that are not growing as much, their growth is blunted. So one hypothesis is, well, the muscles can grow, and that the myonuclear domain is somewhat flexible, but there's a maximum. So there is a maximum level, there's a maximum myonuclear domain that a single myonucleus can support. That's a possibility. Looking at our muscles in the animals, we have come up with a different hypothesis, and that is that the muscles become fibrotic without satellite cells, and that actually physically limits growth. So the muscles become so fibrotic and surrounded with collagen. Here, picrocereus red, and you can see even in the vehicle-treated animals here after 8 weeks, that there is a bit of an elevation of collagen around the fibers by picrocereus red staining, which is dramatically increased in the absence of satellite cells. And here with wheat germ agglutinin staining, which stains glycosaminoglycans in the extracellular matrix, you see the extracellular matrix is really expanded in the absence of satellite cells, which is quantified here. So we've been working under this, thought this was really interesting. And of course, we started thinking about this a lot and thought, okay, well, how might this happen mechanistically? And fibroblasts, there's a lot of fibroblasts in muscle, and they are the primary source of the extracellular matrix collagens and such. And so we thought maybe there was an interaction between satellite cells, and once they are activated and are proliferating, the myogenic progenitor cells, which are the daughters of satellite cells, maybe there's an interaction and there's something going on there that really regulates the ECM. So we isolated the MPCs, the myogenic progenitor cells, from muscles in mice, as well as the fibroblasts. And we did some co-culture experiments. And here's just an illustration, a cartoon to show you what we did, is that we plated the muscle fibroblasts on a plastic well, and then on an insert, we plated either the myogenic progenitor cells or fibroblasts, just to account for depletion of nutrients in the medium, so that there was the same number of cells in all cases. And then we isolated the fibroblasts from both, isolated RNA, and did some real-time PCR. And what you can see, the co-culture with myogenic progenitor cells really caused, across the board, in any extracellular matrix component nearly that we've looked at, is down-regulated, with some exceptions, when you do this co-culture with MPCs, suggesting there's a communication. So to try to get at what that communication might involve, we isolated conditioned medium from MPCs, and we also isolated extracellular vesicles or exosomes from the media of MPCs. And when you put either the conditioned medium or using just fibroblast conditioned medium as the control, but add the MPC exosomes, you see in those cases that compared to just fibroblast conditioned medium, you see a nice decrease in the expression of genes encoding collagens, really suggesting that there's a release of factors from MPCs that are directly influencing fibroblasts. And so when you start to think about inhibition of gene expression, the thing, because I collaborate a lot with John McCarthy at Kentucky, who's very into microRNAs. Of course, that's the first thing you think, okay, maybe microRNAs are contained within MPC EVs, extracellular vesicles or exosomes, and are delivered to fibroblasts and may inhibit gene expression. And so the first very simple experiment, but I think is very visual, that we did is when we isolated the EVs from, I'm using too many abbreviations, EVs from MPC-CM, we just incubated them in acridine orange, which is a dye that intercalates into RNA. And then we wash those EVs extensively and then put them on fibroblasts. And what you see is that the fibroblasts take up, there's a lot of RNA that's labeled and it's taken up by the fibroblasts. So we really, our data are suggesting that it's microRNA delivery via EVs that is influencing fibroblast gene expression. And I'm not going to go into any more detail. This study was published almost 3 years ago now, in which we drilled down and looked at targets in fibroblasts, identified RRBP1, which is a master regulator of collagens that is targeted by NER206, which is the most abundant microRNA in the MPC EVs. And so we really think there's a lot of communication between satellite cells or MPCs and fibroblasts during hypertrophy that really regulates the extracellular matrix. So, but what we wanted to do then is to try to come back to this, okay, is it fibrosis or is it a myonuclear domain sealing that is limiting hypertrophy in the absence of satellite cells? And what we did is we went back to our PAX7 CRE-ER driver mouse, but we crossed it to a different mouse to try to directly test that hypothesis. And what the mouse that we crossed it to is, it's a mouse in which a gene that is required for satellite cell fusion, a gene called NWASP, which is involved in actin skeletal rearrangement, and it's been shown to be required for satellite cell fusion into myofibers, is floxed, okay? And so that when you cross these mice and do the tamoxifen treatment, the satellite cells are still there. It doesn't kill them. All it does is it recombines out. So the satellite cells have the NWASP gene deleted. And so they, to inhibit their fusion. I should say at the same time, the recombination event activates a reporter gene, TD tomato, which causes all the satellite cells to be red. And so here's just a picture of an isolated myofiber, and you can see the nice, beautifully stayed red satellite cell. And here are just some images. I hope you can see them, just dabby stained, but the PAC7 immunohistochemistry nicely lines up with the red TD tomato, and here just merged. So the idea was the satellite cells would still be there, but they couldn't fuse, so we could test this hypothesis. And we felt like this was going to work because if you take MPCs out of the animal and treat with them with tamoxifen or vehicle, see, normally we get beautiful fusion in these big, nice myotubes in vitro, whereas in the tamoxifen-treated animals, fusion is severely impaired. So we thought it was worth going ahead and trying this experiment to see if we could tease apart the myonuclear domain theory versus fibrosis. And here's the experiment, just the design. Again, we did the 5 days IP injection of tamoxifen or vehicle. We did a 14-day washout, and this is in mice that are greater than 4 months old. And then we only let them go 7 days. This was sort of a proof of principle experiment. We sacrificed them. And what you see is that we got nice hypertrophy in both cases. So the deletion of this gene didn't seem to interfere with hypertrophy. You see that we saw a nice increase in satellite cell abundance here labeled in pink. So that the satellite cells were increasing with 7 days of overload, which we expected, and myonuclear accretion was inhibited. So here you can see normally during 7 days, the cells have started to fuse by 7 days, and that was completely inhibited in the NWASP minus TD tomato positive cells. And these are single fibers, isolated fibers where we counted the myonuclei. So we thought this was worth going ahead with the experiment. And so we did a large cohort of mice, and we performed synergist ablation, and we went 8 weeks. And basically the satellite cells, although initially they don't fuse, they eventually catch up. They are able to fuse, so that at the end of 8 weeks of the experiment, there were almost as many myonuclei in the NWASP minus mice as others, so that we in the end weren't able to test the hypothesis. Really disappointing, but I think people need to present negative data, and we spent a lot of time and a lot of energy on this animal, and in the end we couldn't test our hypothesis. We have ideas of other genes we could delete in other ways to impair fusion, but leave the satellite cells there to try to test our hypothesis. But right now I can't tell you the answer, if it is a myonuclear domain sealing or fibrosis. But again, paying attention to this 7-day experiment, though, we noticed some very interesting things. And so always keeping your eyes open and being curious and following up on observations. And that is, with 7 days of overload, there's no myonuclear accretion. There's still red muscle fibers. Not many, but there are some, and some are very dim. You can see the brightly stained satellite cells here. We were really shocked, and we repeated this many times, and there are clearly, and even at 4 days, when there's really no fusion, even in wild type animals yet, we don't detect fusion until 5 or 6 days, you see red fibers. So this really surprised us, but of course it got us thinking about extracellular vesicles coming from MPCs, and could they be delivered to myofibers? And what we found is when we isolated the extracellular vesicles from these animals, that in fact these extracellular vesicles are packed full of TD tomato messenger RNA. And in fact now more recently we've shown that the EVs are actually read by high resolution spectroscopy, as well as nanosite fluorescence detection of particles, extracellular vesicle particles are red. So the satellite cells, for some reason in the MPCs, package TD tomato message, this reporter, into EVs and release them from the cells. And so because we saw this, we did an in vitro experiment to follow up our observation on these red fibers in vivo. So we isolated these EVs and put them on in vitro into the media of just wild type black 6 primary myotubes from black 6 mice, C57 black 6 mice. And this is what we find. Here are the TD tomato negative EVs, so from the vehicle treated animals. So here are the myotubes just for black 6 mice. And here they are nicely stained with DAPI, and this is myosin. What you see in those that are from the NWAS minus TD tomato positive EVs, I hope you can see the red speckles, that those EVs are delivering TD tomato to the muscle fibers. And I've shown a higher MAG here, just hoping that you can see that the myotubes in vitro are red. They're taking up TD tomato. So this has opened up sort of a new avenue for us and a new tool, because we are beginning to explore now in a variety of cell types. You know, satellite cells or MPCs can communicate not only with fibroblasts and now apparently with the fiber itself, but with other cell types. So we've begun sorting for TD tomato in muscle, all different mononucleated cell populations, and performing single cell seq to look at how those cells that have received EVs versus those that haven't, how their gene expression is different, and what microRNAs might be delivered. So I had hoped we would have some data that I could show you today on that. I don't, because we're just getting into this single cell RNA-seq, and it's more complicated, but we're very excited about it, at just using this TD tomato reporter mouse as a way to track MPC EVs. And my postdoc, Kevin Murrock in the lab, put this slide together, which I really like. He spent some time. And so if this is the satellite cell, what I've just told you about our most recent work, which was just published a month ago in the new APS journal Function, can communicate with the muscle fiber, and almost certainly the fiber is communicating back to the satellite cell. But our work and others, I've told you about our work showing that satellite cells communicate with fibroblasts. Many other labs have shown communication to the motor neuron, endothelial cells, and macrophages. And I want to follow up on this just for a minute, because we're getting very into macrophages in the lab. And they've been very well studied within the context of regeneration, but not so much with hypertrophy. Benedict Chazot has done a little bit of work, but we were really interested in the role of macrophages in hypertrophy, because they are so abundant, resident macrophages. And these are ones that express primarily M2 markers, that is, markers of anti-inflammatory reparative macrophages. These are really quite abundant in muscle, more abundant than satellite cells. And here's just some staining with a PAN marker, 11B versus 206. And you can see just in this slide, here's one macrophage, which is not 206 positive. By and large, the resident macrophages are much more, M2 macrophages are much more abundant in muscle, in resting muscle, and those preferentially increase with hypertrophy. And these are data from that we published last year from a human exercise, a stationary ergometry study in humans, which really has piqued our interest and wanted us to follow up more on, made us want to follow up more on macrophages. You see these M2 macrophages increase with this training paradigm. They're correlated with the increase in CSA in people in response to progressive resistance cycle training, and that they're very closely, or they're correlated to the increase in satellite cells. And I just wanted to show you this one image where DAPI stains, so you have the two nuclei. This is a macrophage, the one here staged with PAX7 is green, and you can see how closely associated these cells are. And this is very common in the muscle. In cross sections, you see that macrophages and satellite cells are very closely abutted, so that we do think there's lots of communication between them. And more recently, we just completed a large clinical trial, resistance training clinical trial, actually looking at adjuvant metformin in its ability to augment growth, which we recently published, and I'm not going to tell you about that clinical trial at all, except these results, which show that, again, now with progressive resistance training, the M2 macrophages increased, and that increase in macrophages is nicely correlated to increase in CSA. And we believe this may be partially mediated through communication with satellite cells. So that was just a little bit of an aside. And what I want to now just summarize the hypertrophy story, and that what we think is that satellite cells are necessary for ECM remodeling. And this is largely, we think, through cell-cell communication, which may be much more widespread than we had ever believed. So what I want to now talk about is this role in muscle and homeostasis. And I have here unknown, because there are indirect evidence, and most people assume that during the life of an animal, satellites, myonuclei die and are replaced just randomly with a satellite cell nucleus, and that satellite cells are involved in muscle maintenance throughout the life of an animal, which makes sense because it's hard to imagine, you know, that these that the muscle fiber, which is so long-lived, that the nuclei are as long-lived as well. But we wanted to test this hypothesis using our PAC7 DTA animal model, because originally we hypothesized that this would be a model of premature aging. And so here's the design of the study, where we basically took 4-month-old mice and treated them with tamoxifen, as I've told you about. And then we, in our young animals, in all the slides I'm going to show you, the young animals were just killed a month later. So they were 5 months old approximately. The other animals were just allowed to live in their cages for 20 more months. So here you go out to 20 months, a small subset, we injected barium chloride into the tibialis anterior to determine if satellite cells were still depleted and if regeneration was impaired. And the rest of the mice all those mice, together with the rest of the mice that weren't injected, were killed at 24 months. And in all the slides that I'll be showing, 24-month-old mouse is called an old mouse, and it's essentially lived 20 months with no satellite cells, okay? So we were hopeful and were pleasantly surprised, and surprise is the bigger word, that the satellite cells throughout the life of the animal remain significantly depleted. So you can see here that just in the vehicle-treated animals, one loses muscle, specifically satellite cells, with age. Some muscles, not so much. Other muscles, like the plantaris, 50%. And you see that in our tamoxifen-treated animals, we get depletion initially for 5 months, and then at 20 months later, significantly depleted, far lower than anything that occurs during normal aging. And I have to say, I'm still stumped by this, with satellite cells supposed to be the stem cell. Why they don't repopulate, I don't know, but they don't. And so they remain depleted throughout the life of the animal. And of course, what we hypothesize is this would cause sarcopenia. I mean, every grant I review on muscle aging, that's loss of satellite cells causes sarcopenia. Well, here, just the proof of principle in terms of the TA. These are ones that are injected with barium chloride, and you can see that the muscle is regenerated. This is an old animal. Regeneration is impaired. You see regenerated fibers, but you see a lot of infiltrating cells. So regeneration is impaired with age in our vehicle-treated, just normal 24-month-old mice. But you see it's completely abolished. There's no muscle regeneration going on in those animals that lack satellite cells. However, sarcopenia is not affected. So you see that muscle mass, muscle cross-sectional area, fiber cross-sectional area is lower in the older animals. In all cases except the soleus, doesn't seem to lose much mass, and it's not affected by the depletion of satellite cells. So these were very surprising results and were very difficult to publish, and we had to do hundreds of mice and repeat and do all kinds of assays because it's a negative result, and it's not I think, what the satellite cell world wanted to hear, but it's the results. And we were surprised, but it really doesn't exacerbate sarcopenia. We've also done the experiment and killed mice at 18 months, thinking that perhaps it would just accelerate, and it doesn't. So it doesn't really have any effect on sarcopenia that we can detect, except, well, including looking at single fibers. There's no difference in myonuclear density in the fibers, but there is more fibrosis. So in these animals, it's not tremendous, but they're definitely, they have more extracellular matrix surrounding their muscle fibers. So this was surprising again, but many of the reviewers and people that when I present this, they say, but those are sedentary mice. They sit in their little shoe boxes their entire lives. What if they were physically active? Then you would see an effect of satellite cell depletion. So we went back and we repeated the experiment. This time, just because of the mice that we had on hand, we treated the mice with tamoxifen at 5 months old. And then at 7 months, so we had a 2-month washout from the tamoxifen, we just gave them access to running wheels, either locked or running. So allowed them just to be as active as they wanted to be. And we tracked their activity using clock software. So we know exactly how much they ran, and black 6 mice and these DTA mice really like to run. So, they run 10, 12 kilometers every night. And we let them do that. And then we sacrificed these mice at 20 months. So not quite as old. And I'll tell you why we stopped the experiment at 20 months. And here, just first to show you again, even with running, the satellite cells stay depleted for this 13 months of running. Their increased running causes in the satellite cell plus, those animals with satellite cells, running increases the abundance of satellite cells slightly, but no effect. The satellite cells remain depleted in those animals that were treated with tamoxifen. And that you see that myonuclear accretion occurs. The muscle fibers, I'm going to show you this in a minute, do hypertrophy some, and there is some myonuclear accretion, at least in the soles. See, it doesn't reach significance, and we've done more animals now, and doesn't appear to be any myonuclear accretion in the plantaris over the course of this 13 months. But you see that that both using just dystrophin DAPI, as I've shown you, you see a nice increase in the runners, which doesn't happen in the depleted animals, and also using a marker, PCM1, which is a stain that stains the nuclear envelope, which is shown here, specifically in myofiber nuclei, is just another way to confirm that and count myonuclei more reliably. And here's the hypertrophy data. What I broke it down by fiber type because it complicates the story a little bit. But again, the same color scheme where you see in the plantaris, in all cases, that the runners increased fiber size in all fiber types, 2A, 2X, 2B. But I'm not showing you the fiber type data. There was a shift to more type 2A fibers with running, and those are smaller. So the mean fiber cross-sectional area wasn't different. But if you look by fiber type, the fibers all hypertrophy. And this is more apparent, reached nearly significance in the soleus. Type 1 fibers for sure, very close in the type 2A fibers with running, and that this is impaired. So here, hypertrophy in response to lifelong running, this increase in fiber size, was significantly impaired in the absence of satellite cells. So I want to conclude just briefly. I'm here, and I have just a little bit more I want to tell you, but from here, the mouse work, I just want to conclude that regeneration and hypertrophy have different satellite cell requirements, and compensatory mechanisms exist in adult but not growing muscle to enable hypertrophy in the absence of satellite cells. Satellite cells repress ECM production in fibroblasts through EVs, which may facilitate muscle remodeling. Satellite cells depleted muscle becomes fibrotic over time, and this may impair growth. Again, I can't address the myonuclear domain theory at this point, a lifelong depletion of satellite cells in sedentary mice that impairs regeneration without affecting sarcopenia. And from those studies we did, and I'm going to show you just a little bit of work from humans, satellite cells may contribute to growth via different mechanisms, with oxidative muscles having more dependence on fusion, and that we think that perhaps loss of satellite cells with age may limit muscle adaptations to physical activity. So they may have more of an effect on people who are really physically active, this loss of satellite cells. So what I'm going to end with, just a few slides. I want to show you some of our work that addresses satellite cells and this contribution to hypertrophy in a fiber type specific way in human adaptation. And this is, I'm going to show you some work that was published several years ago in humans to just look at, again, this was some of the study I told you about with cycle training, progressive resistance cycle training in people. And you can look at the different fiber types, and that we really find that, as many others have, that the abundance of 2A, 2X fibers, which these hybrid fibers are really a sign of a sedentary, inactive lifestyle, and those increased with BMI, and that looking at fiber type, we see again a shift in fiber type, where you see an increase in type 2A fibers with training, and a decrease in these 2A, 2X hybrid fibers, no change that we see in the type 1 fibers. And that we see that there is a modest increase in fiber CSA in these people with the cycle training, as I've mentioned before. And here, just binning the fibers by fiber size, you see that pre-training, there's a higher number of the smaller fibers, and the curve is shifted to the right, showing that there is this hypertrophic effect. Now, what we found is that both type 1 and 2A fibers hypertrophy, no significant hypertrophy in the 2A, 2Xs, which, and they were also reduced in abundance with age. So both type 1 and 2A fibers increased in abundance. And now let's look at satellite cells, or increased in size, sorry. They both showed fiber hypertrophy. We wanted to look at satellite cells, but here in human, it's really easy. Staining works so much easier in mouse because many of these antibodies are mouse monoclonals, which are tricky on mouse tissue. But here is a type 1 antibody, type 1 myosin heavy chain. This is laminin, just to show the fiber borders, satellite cells, and then DAPI. And if you overlay them, what this allows you to do, because laminin, you know, is in the basement membrane, and the satellite cells are underneath the laminin, very close to the muscle fiber. So it allows us to assign a satellite cell to a type 1 or a type 2 fiber. And that's shown here, a little bit blown out, but here clearly is that's a type 1 satellite cell, and that's a type 1 satellite cell because it's underneath laminin, closely associated with this fiber, whereas the laminin goes on this side. So this is a type 2 satellite cell. And what you can see is that satellite cells increase with training, and this is preferential to type 1 fibers. We saw no increase in satellite cells in type 2 fibers, and further, myonuclear accretion was preferential in type 1 fibers. And I think this is important because I want to circle back to our synergist ablation model in the mice, where you know we're limited to look at just the plantaris. And the plantaris is a fast muscle. So it's almost exclusively type 2 fibers. And we show this flexibility in the myonuclear domain, and the fibers can grow even in the absence of satellite cells. We're beginning to think that perhaps the myonuclear domain is more flexible, much more flexible, and there's data out there to suggest that in a type 2 fiber, whereas type 1 fibers, oxidative fibers, may have a more stringent requirement for satellite cell fusion to grow because they're so much more metabolically active, they have more mitochondria and such. So with that, I'm going to conclude just this little section on humans, in that human muscle, type 1 muscle fibers have a more stringent requirement for satellite cells for growth than type 2 fibers. And so I want to say in our mouse models, we've now switched over, and we've published a couple papers on this new model we're using instead of synergist ablation, called progressive weighted reel running. We call it POWER. And it really is a nice hypertrophic model. It does combine aerobic and resistance training, but the muscles hypertrophy nicely, and that doesn't limit us to the plantaris. So we can look at all muscles in the hind limb, and it will start to allow us to look at the response in a fiber type specific way and a muscle specific way, because I think that different muscles have different satellite cell requirements, which brings me to my take-home messages, and that is that the necessity for satellite cells for hypertrophy, I don't think one size fits all. I think it's very fiber type and muscle specific, that different muscles, because I don't necessarily think a 2A muscle fiber in the soleus is the same as a 2A muscle fiber in the plantaris, in a primarily fast muscle. And so I think it's going to be interesting to really tease that apart, that it's dependent on age. So young, very young growing animals absolutely require more satellite cells to grow. And our hypothesis, they're at their max, their transcriptional max, because they're growing. And in order to grow more in response to a hypertrophic stimulus, you have to give them an extra nucleus. And then finally, the mode of exercise. I've talked about several different modes of exercise, synergist ablation, a surgical model, cycle training, resistance training, just lifelong running, and now power. And I think these all are going to have an effect, as is even just in these surgical models in mouse, the different ways to hypertrophy your muscles. I think they're going to have a big influence on whether you need satellite cells or not. But one of the first, the biggest, and our newest avenue, and we're really going down this pathway in a big way, is the cross communication between satellite cells and other cell types in muscle may regulate the muscle microenvironment, its adaptability, and ultimately physical function. So that really we think satellite cells, activated satellite cells and MPCs, play a much larger role in adult muscle adaptation to exercise training than simply providing nuclei to growing muscle fibers. And with that, I'm going to give credit where credit is due, that a lot of the work I've told you about started several years ago, and people have left the lab, but I still want to mention them. They're all still in science, either have started their own labs or were graduate students in my lab and are now postdocs, or several of the students have gone on to graduate school. So I really want to thank particularly Chris Fry, who is really instrumental. And these are current members of the lab that participated in this study. Kevin was a big part, as was Corey and Kate, but everybody played a big role. I, of course, want to mention my collaborators at UK and within the Center for Muscle Biology, John McCarthy, Esther DuPont-Versteegden, and Phil Kern, who's our study physician and does all our muscle biopsies and really helps with our clinical trials. And then a lot of the work on our clinical trials we do in 2 site centers collaboratively with Marcus Bamman at UAB. And then, of course, my funding sources. So with that, I'll stop and be happy to take questions if that's a possibility.
Let's hope it's a possibility, because I am sure that there's going to be lots of questions for you, Charlotte. What a wonderful talk. Thank you. So rich in information for us. Wonderful. I got lots of questions myself, but folks, please ask your questions through the Q&A panel, if you don't mind. And my job is to relay the questions to you, Charlotte, so that you don't have to read those questions. And so my first question is, and then I'll take others' questions as well. I'm compelled with this communication thing that you've talked about between the satellite cells and other cell types. Macrophages seem quite interesting, but you mentioned the type 1 fibers and having higher mitochondrial content and oxidative capacity. And so my first question is there some communication between a satellite cell and something in a type 1 fiber that has more mitochondria, rich in subsarcolemmal mitochondria, for example, and just wondering what that connection is. And so good that you did the human study to reveal that, I guess.
Yeah, that's such a good question. And, in terms of, I can see that you need a satellite cell to fuse in, to donate mitochondria plus the nucleus to generate more of the, components of the mitochondria, so you can make more mitochondria. In terms of just the EV communication, I have less ideas on how that might be affecting mitochondria, and particularly subsarcolemmal mitochondria. But it does make me think, there have been a couple papers, mostly showing in vitro, but some in other cell types, these tunneling nanotubes. And people are really interested if the satellite cell, because it's right up against the muscle fiber, if they're tunneling nanotubes and the satellite cells actually deliver mitochondria to them, or, mitochondria can go back and forth. So I think there's lots of ways potentially for communication there, but we think of it more as that, you know, in terms of growing, and that there the myonuclear domain maybe is more fixed because the nuclei are just cranking out because they've got to produce all these components of the mitochondria, and they're just so much more metabolically active, and that you need just another nucleus to fuse.
Yeah.
Fiber type specific communication, we haven't really gotten at to myofibers, but it's an interesting question. I'll have to give that some thought.
Yeah. No, I mean, if the satellite cell can donate a nucleus, it can also contribute mitochondria, and perhaps, you know, there's some interaction there.
Yeah.
Okay, so Dr. Hawk from McMaster is asking, what are your thoughts about myofiber splitting? Do you think that myofibers that lack satellite cells are more likely to split to maintain myonuclear domains, and thus may account for some hypertrophy findings?
And I want to say hello to Tom. I'm sorry I can't see him, but you know, hello. And yes, you know, Kevin Murrock in my lab published a short review on fiber splitting, which actually in the synergist ablation model is fairly common, because although fibers hypertrophy, I think the increase in CSA would be more, but the fibers do tend to split because we see not only larger CSA, but more fibers, the number of fibers. Because in our mouse studies, unlike in human, we count all the entire cross section, and you count and you can tell there are more fibers with synergist ablation. And I don't think we don't have signs of a lot of regeneration or new myofiber formation. The fibers are all sort of bigger, and so I do think they split. And Tom, I do think, yes, we haven't quantified it carefully, and whether the lack of satellite cells causes more splitting. I'm trying to think if in any of our studies we show that there's more fibers in a satellite cell depleted muscle than not, implying the existing fibers are splitting. I'll have to go back and look at that, and Kevin may have commented on that, but there is, he published a review where he really summarized in that this idea of fiber splitting is actually potentially more common than we think, and it may actually be exacerbated by satellite cell depletion. And I don't know that we have really quantitative data to show that, but I think it might be the case.
I am surprised to hear you say more fibers, because I go back in history to Gagne and Gulnick and those people who counted muscle fibers with tenotomy or overload and really didn't see any, but maybe newer technology allows us to better do that.
Yeah, it's definitely, there are more fibers. And even with our power model of running, you know, with this resistance training where they're really pushing hard on the wheel to run, so there we see nice hypertrophy, but there's also more fibers. I mean, so we've got to follow up on that in more detail, because I know that, you know, with this fibers hypertrophy, there's no hyperplasia. But I do think splitting actually is a bigger thing than I would have thought. And yeah. I wish it didn't exist, but I think it does.
Okay. No, you're the expert. Dr. Tsushima is asking, in your satellite cell minus mice, were there any changes in whole animal activity, like distance on wheel running or muscle function, force generation, anything like that?
In some of the experiments, yes, and some no. And in those, the one that I forgot to say, in that the last set of experiments in the mice where we let the mice have access to the wheel for most of their lives, and we killed them at 20 months, it's because they started not to run. Their running volume was declined, and the decline was greater in the satellite cell minus mice. By the end, they just weren't running much, and we were worried we were going to lose the training effect, so we killed them at 20 months instead of going out to 24. And what we think is happening, and we published a paper, Esther Dupont-Restigden was the senior author on it, in our animals, the animals without satellite cells, especially if we deplete satellite cells and then give them a long washout, a month or two, and start them running, they run slower and they're uncoordinated. It's because they're wheel running, they run slower, and they fall, and we've done balance beam and they fall off more. And when we look at the muscles, The spindle fibers, the interfusal fibers in satellite cell minus mice are buried. There's so much more ECM and the lobes are smaller. So we actually think they have a proprioceptor problem, which means, they can't find their position in space as well. They're less coordinated, so they do run less. We tend to, now we've realized that, and if we start them running within 2 weeks after tamoxifen, we see less of an effect as if the running prevents some of that fibrosis, but they do get fibrotic. And we think that has a particularly negative effect on proprioceptors, the spindle fibers, which have implications for aging. It's why people maybe don't have as much balance and coordination. If their satellite cells are going down in number and they're becoming and surrounded by collagen and things, that it may make older people more prone to falls.
Okay, fascinating. I admire this animal model that you have. It's just must have taken so long to generate, and you've used it so well. Is it a C57 Black 6 background?
It is.
So one question has come up by Dr. Chris Perry, asking about if a different mouse strain, which may have a lower regenerative capacity, do you think comparing mouse strains could be another approach? I mean, and might give a different result. I don't know. What do you think?
Yeah, I mean, there's so many differences between strains. We've talked from time to time, we wanting to go into the 4-way cross to make it more, more like wild type, because there's such vast differences between the strains. And I do think it would make a difference. And we're glad we're using Black 6 because they like to run. Some strains don't run. They don't like to run. Black 6 mice do. I don't, how to ablate satellite cells specifically in different strains to see the effects, but there are clearly different regenerative capacities between strains, which are probably related to different satellite cell function or abundance or something. But how to do that experiment is really hard. I mean, I don't know how to do that experiment.
Fair enough. So Dr. Peter Bax is asking, what's giving rise to the compensatory hypertrophy in adult animals, I guess, if it's not satellite cells? How is the muscle growing?
That's a great question. And here's one of our hypotheses right now, is we've done some experiments where we, in either control, sedentary animals, or after running, right about 5 hours, we did a whole titration time course. Shortly before we kill them, 5 hours or so, we inject them with EDU, which is a uridine analog, so it labels RNA. And you can do click chemistry, and you can then visualize nuclei that have undergone a lot of nascent RNA transcription, right? And if you do that in a sedentary animal, only about 25% of the nuclei are EDU labeled. As if the muscle, not all the nuclei are active. If you're in a sedentary animal, only a subset of your nuclei are actually very transcriptionally active, and EDU is probably mostly a marker of ribosomal RNA because that's mostly what's being produced. And most of the nuclei are not labeled at all. And then when you do synergist ablation, and you look now up to 40 or 50 or 70%, I can't remember the exact numbers, of the nuclei are now EDU labeled. And so you've recruited nuclei that weren't active before. Well, I really believe that your myonuclei are not at their transcriptional capacity, and that normally you fuse in a satellite cell and it kind of spreads the load, okay, in the same proportion, and they add something and you're good. But if you don't have satellite cells up to a point, I think the existing myonuclei have a lot of transcriptional reserve capacity, and they can support a lot of growth up to a point, probably, without satellite cells. And we're trying to design some experiments to test that. But I really think that your myonuclei, even under hypertrophic conditions, and what we've shown is with these EDU experiments that, okay, at 7 days of overload, or early on, 2-3 days of overload, you see this huge increase in the number of EDU positive nuclei, which by 14 days decreases, because I think it's now translational efficiency and things, and maybe post-transcriptional regulation and stability of messenger RNAs and things. Except in the satellite cell depleted muscle, that transcription stays high at 14 days. So that's what I don't have any really hard evidence to show that, but that's what I think. There's a huge transcriptional reserve capacity in existing myonuclei, and that they can support growth without satellite cells.
Yeah, I think that's a great answer. So Dr. Shime is asking, what do you think that there are different oxidative capacities, for example, in satellite cells associated with different fiber types? Like, so satellite cells associated with slow fibers or fast fibers, do you think they have different oxidative capacities? The satellite cells themselves, I guess.
Yeah, well, you know, quiescent ones are so quiescent and so transcriptionally inactive and metabolically inactive, but once they're activated, you know, there are evidence, you know, that satellite cells are different, that a satellite cell knows if it comes from a fast fiber or a slow fiber. Old experiments back from Simon Hughes and others. And, you know, I'm trying to think, there is evidence, and I'm trying to think, I think it's Victoria Sartorelli has actually shown that they are different, that you can promote, sort of like with macrophages, where inflammatory macrophages are more glycolytic, and then when they convert to M2, they become more oxidative, and that really is a big part of their differentiation. So I think probably so, and I could recommend Vittorio's work, Sartorelli. He's at the NIH, the NIMS intramural program, and I think he has actually shown that, and I don't know, it may just be activated versus quiescent, but certainly the metabolic activity of satellite cells changes and is different. So I think that's entirely possible.
Okay, good. 3 more short questions I have here. Let's see, keeping you on the hot seat, Charlotte.
This is fun.
Yeah. So Catherine is asking, and surprised, I guess, at the fact that, you know, if you deplete satellite cells that early in life and let them go for the 20 months, for example, why is it that they don't regenerate that pool? How come is that? Why?
It has, and we've tried a couple of experiments or a couple of mouse strains to try to get at that, and I don't know, it has me totally stumped. But here's my latest hypothesis, and it's based on Sherigan Tagbahash's work, where he has shown, you know, people are showing more and more that, and by single cell seq, and even before that, that satellite cells are heterogeneous. So you can look at the PAX7 positive cells and you can isolate them, and people have shown they're actually heterogeneous. And what Sherigan showed a number of years ago, his identification of satellite cell heterogeneity was there were PAX7 high cells and PAX7 low cells. And that he showed in vitro they do different things, and I can't remember exactly what he showed, but that he thinks they're different, and it's the PAX7 high cells that are, and they're fairly rare. Most PAX7 cells in the muscle are lower in their expression, and he speculated, his data suggested the PAX7 high cells were the bona fide stem cells, and the others were more progenitors, you know, not, well, since what we're killing the satellite cells for is using a PAX7 promoter to drive CRE and DTA, that we're preferentially killing all the PAX7 high cells and therefore depleting the bona fide stem cells. And what you have left, they can divide some, but they're not stem cells. They're the progenitor cells. And trying to design experiments to try to test that idea, but that's the way I'm thinking now, is that we've somehow preferentially, with our model, inadvertently, preferentially killed the subset of satellite cells that are the bona fide stem cells.
I see. Okay, so related to that then, Dr. Johnny Parise from McMaster is asking about, does the loss of satellite cells in the model alter the proportion of other cell types in the muscle? So other progenitor cells, I guess, so it's kind of related to what you've just said. And would those, if there's some compensation there, would that be important in signaling? I don't know.
Yeah, and hello to Gianni as well. I wish I could see you all in person. But yes, fibroblasts increase in the absence of satellite cells. FAPS, we're doing single cell seq now and showing the changes in the different populations. But yes, I think they do change, and their identity changes, that we've got some very recent that I don't know quite what to make sense of, that the fats may even become preferentially, they differentiate more rapidly, and doing RNA velocity and trajectory analysis on the single cell seq data, and they may even take on more of an immune function in the absence of satellite cells. So I think the existing populations change, their relative abundance and their gene expression profiles and what they're doing change a lot. And just to follow up on that, this makes sense in my mind, because when we do this tenotomy or synergist ablation, the increase in satellite cells, I mean, they increase by 3-4 fold. I mean, you know, there's a huge increase in the number of satellite cells. Well, not all of them fuse, for sure. You don't, not anywhere close to all of them fuse into the fiber. So what are they doing? And it makes so much sense that they're conditioning the environment, they're communicating with other cells to facilitate growth, and then they die or do what they do, and that they change a lot of things, and that's a big part of their function. And only a small subset fuse into the fiber, and a small subset go back to being stem cells, and you know, the rest apoptose, or you know, but that they have a big function in controlling other cells.
Right. Final question. So I wondered about the work that you're doing and the relationship to what Carl Gunderson shows in Oslo. You know, he talks of, you know, the muscle memory things and the long-lived nuclei and the fact that they remain during atrophy, and the accumulation of myonuclei during hypertrophy, I guess, similar to what you show. Does your work and his work agree? Do you agree with the thinking that Dr. Gunderson presents? Do you have any comments on that?
Sure, and a lot no, we don't agree, because and a lot of his models are, you know, these denervation, and they're very and surgical, and so they're not really training and detraining. They're, you know, denervating and then adding testosterone and doing these funky things. Now we found too that if you atrophy muscles in the mouse, we don't lose myonuclei either, and the fibers regrow without. In the rat, we lose nuclei, and I think it's up in the air in humans. You do bed rest studies have been long enough. Some show you start to lose myonuclei, others show you don't. So with that, with atrophy, I don't know, but we just published a paper in the Journal of Cancer, Sarcopenia, and Muscle, or that journal, JCSM, where we, with our power model, we did extensive, trained the animals, they hypertrophied nicely, and then we detrained them for long periods of time. So and the plantaris, the gastric, they gain nuclei and they lose them with detraining. The soleus doesn't. It gains nuclei and it keeps them, but that's probably, you know, it's a tonically activated muscle. It's different, and the animals tended to gain weight, so maybe, you know, they were more chronically loaded. But I think there's muscle memory. I think it has nothing to do with the number of myonuclei you have. We've also published a while back in APS, I think Tom Hawk was the editor actually, where testosterone treatment, we used our DTA mouse model with testosterone, a vehicle gave them testosterone, and you see nice hypertrophy in both. And that's not dependent on satellite cells. And we've now more recently then removed the testosterone, and they go back to normal size and they lose nuclei. So I don't think it's a numbers game, and certainly with training and detraining, then if you retrain them, and Kevin has an experiment going, you know, where he trained the animals for 4 months, he'd retrain them for 6 months, and now he's retraining them. So, you know, 2 years later, we'll have the results. But what is the basis for your ability to gain muscle more after you've trained, and then you stop, and retraining everybody knows you feel like you can, you know, you gain muscle more quickly. I think it's epigenetic. I think there are changes in the nuclei that may be maintained epigenetically, but I don't think it's a numbers game. I think it has nothing to do with the number of myonuclei, and I think the data there have been way overinterpreted. And if you look at the models, they're so artificial in terms of what they did to the muscles, that in terms of training and detraining, and this power model is really allowing us to train them, detrain them, retrain them, and we can get to the bottom of it. But it's looking like it's going to be different by muscle, but for the most part, it's not dependent. Myonuclear number isn't what's doing it. I think it's epigenetics.
Wow, fascinating. I'm really looking forward to seeing that paper come out because we don't agree, he and I either. So anyway, it's fascinating. You know what? We've asked you lots of questions now, Charlotte. It's time to stop. And for those who have submitted a few additional questions, just I'm sorry that I can't. We've asked plenty of questions. So Charlotte, please stay with me though, and I want to thank you so much on behalf of the Muscle Health Research Center for accepting our invitation.
Oh, no, this is great. And I really hope, I know you were planning tentatively a meeting next year, and fingers are crossed that we can do it. And because I really want to get up to Toronto and see you all in person and stuff. So I'm hopeful. Maybe we'll get to do this again in person.
I trust that we will. I believe that we will.
I'm hopeful, you know, guardedly optimistic.
Right. Okay. So thank you very much. Please hang out.
Okay, I'm here.
Yep.